Thank you for the introduction. I just want to start by saying this is a really great opportunity for me, so let's keep it about me. For someone like me to be able to speak in front of an audience such as this, it's rare that graduate students get the chance to have invited talks like this, and also be able to disseminate high-level biochemical information to an audience that maybe doesn't always see something like this. So when I started with this fellowship with the PSSO, I wasn't working on anything to do with Parkinson's disease, but it did turn into a project with Parkinson's disease when, as we were reading the literature, it kept coming up time and time again that one of these proteins that we're interested in that I will get to is probably related in some way, but there's not much known about it. So I just wanted to start by mentioning probably something that most of you know much more than I do about Parkinson's disease is some of the projections of the disease itself. It seems to be accelerating at quite an intense rate. We're expecting about 65% increase of Canadians developing Parkinson's disease by 2031, and certain other things like the cost of care, about $1,100 on average per year. When I saw that number, I was actually a little bit surprised by that. It seemed actually low to me, and I would expect in countries maybe like the USA for that to be much higher actually. And then stress levels. So when you're dealing with this type of disease and caring for someone, for yourself, the stress levels, according to certain studies, double, whatever that means. I'm sure people dealing with that disease can speak to that much more accurately. So the PSSO, in collaboration with MyTax, funded 7 graduate research projects this year. So you guys are stuck with me today, but there were 6 other students that were also awarded these stipends. And it seemed like the PSSO did a really good job at selecting quite a broad category of students from what I do, molecular research, to the diagnosis of disease, and all the way to the maintenance of it. So they really covered a spectrum of outcomes when it comes to the research of Parkinson's disease. So I'm just going to first start my talk by mentioning PD and the molecular details of it at kind of a medium level, before then going into what I actually been working on for the past year. So the signatures of Parkinson's disease, again, this is something that I think most of you probably understand already. There's 2 ways you can look at it. The molecular signatures, which are the loss of dopaminergic neurons in the brain, SN region being the substantia nigra. And then you have these neuronal inclusions, these Lewy bodies that are really the root cause of these lost neurons or these dying neurons. And these are alpha-synuclein positive, which is maybe a protein that you've heard about before. And then the symptomatic signatures are shown as well. These are mainly related to the motor defects. So a question I get a lot, whether it's to do with Parkinson's disease, cancer, anything that biochemists are researching is there a cure for this? Is there a cure for that? Or why is it taking so long? And quite frankly, the reason is because these disorders are extremely complex. And so PD is definitely probably one of the more complex ones as well. And so we ask ourselves, why is something like Parkinson's disease so intractable? Why is it so hard to treat? And there's many reasons, and I'm just going to summarize a few of them, and I'm sure many physicians can add even more to this. So you can look at the onset of the disease. So what are the genetic factors, the environmental factors that go into it? And so, for instance, you have quite a laundry list of susceptibility genes that can be indicators of the onset or progression of Parkinson's disease, and those are listed there. And so these are some genes that can be mutated over time, inherited, and these can lead to different aspects of Parkinson's disease. And whether or not environmental factors actually influence the genetics is another aspect that scientists don't fully understand yet. Another thing I want to mention is that about 90% of Parkinson's disease is sporadic, which means it occurs over time. It's not inherited. There's no genetic link to it. And that actually makes it more difficult to track the disease and understand how to treat it or how to detect it before it even happens. And another thing to mention is that we lack preclinical models. So when we are studying the disease in the lab, how do we recapitulate it at a lab scale in a way that can be as accurate to the biological scale that we see with aging adults? And that's something that we struggle with. So whether it's selecting mice, rats, who knows what you could be using. Again, this is research I don't actually do myself, but there are many model systems you can choose from, and they all have their pros and cons. And what is consistent amongst these models is you are giving up a lot of biological relevance to try and simplify the disease as much as you can so that you can actually make it something that you can study in the lab. And so it's great because you can make progress over time, but it makes something like this take a very long time, which is why despite the decades that Parkinson's disease has kind of been under the microscope and the amount of money that has been put into it, we haven't yet made major strides forward in terms of being able to stop the disease in its tracks or detect it before it happens. So other aspects that make this difficult is diagnosis. So classically speaking, Parkinson's disease has a very long prodromal phase, which is a fancy way of saying the phase at which the disease is progressing, but clinicians can't detect it, and therefore they can't diagnose you. So within those tissues, within the neurons of your brain, they are being damaged over time, but we don't actually know that that's happening until you start to exhibit symptoms that can be diagnosed, so like motor disorders. And once that happens, it's already pretty much too late because that's when you've had a substantial amount of neuronal loss. And so it's kind of a double-edged sword in that way. There's also extensive genetic and phenotypic heterogeneity. So there's no cookie-cutter aspect of Parkinson's disease. There are a set of parameters by which clinicians may diagnose you based on, again, motor symptoms. But not all these symptoms are the same for anybody with Parkinson's disease. And sometimes you have people with Parkinson's disease and it's on a spectrum with other things like Alzheimer's. And so it just adds to the complexity. Again, we're just talking about how complex this actually is. And that's what we get into clinical overlap, is that you don't have these diseases in isolation often. And so how do you detect them and how do you separate the population and identify who has what? And then we lack specific biomarkers. So no matter what stage the disease is progressing, how do we detect it? And that's something that is, again, a big struggle. So one thing that you can do is when you start, when you have the idea that you are progressing with some kind of disorder, you can ask your doctor for something like a DAT scan, which is a way to measure your dopaminergic activity in the brain, and characteristically you see a decrease in somebody that is suffering from neuronal loss. This isn't something that is specific to Parkinson's disease, however. So that type of imaging is actually not going to differentiate from other types of diseases or deficiencies. There is a lot of research, however, looking at biomarkers in fluids. The cerebrospinal fluid is probably the most popular, although it's a bit invasive. That's why there's even more research looking at blood biomarkers. Some of the big ones I would like to just point out are the early biomarkers that we're trying to get to would be phosphorylated alpha-synuclein, oligomeric alpha-synuclein, and I'll talk about this a little bit more, but this is the protein, again, that leads to those big Lewy bodies, those aggregates that are leading to neuronal loss. And if we can detect a higher level of those in body fluids, they may be an indicator of progressive Parkinson's disease. However, this is, again, very much in its infancy when we think about the actual research stage that it's at. And then a final aspect of the intractability of Parkinson's disease is the treatment itself. So we don't fully understand the biochemical pathways that actually lead to the symptomatic outcomes that we see in the clinic. Again, we understand that these Lewy bodies are causing or leading to, in some way, the degradation of neuronal tissue, but even that mechanism in itself is under question. There's also many targets that could be leading to Parkinson's disease. So it's not just about alpha-synuclein, this protein I keep mentioning, but there are many other ones that lead, that can either feed into the alpha-synuclein pathway or are completely independent of it. And so which ones do we have to target, if not a single one, but multiple? And that's why that idea of a silver bullet, meaning, is there one cause of Parkinson's disease? Okay, let's focus on that and then hit it with our silver bullet and then cure the disease. Or do we need to consider multiple pathways at the same time with combinatorial methods? And then one of the other things is looking at familial versus sporadic PD. So it's the genetic versus the form that is taken on over time, and I'll mention that in the next slide. But the blood-brain barrier is a literal physical barrier to us actually understanding how to treat the disease. So we can make as many compounds in the lab as we want and go through clinical trials, this and that. But if we can't get the compound out of the bloodstream into the brain, it's not going to do what we need it to do. And that is a significant barrier because of roughly 99.8% of small molecule compounds are filtered at that stage. So they can't pass that blood-brain barrier. And that's beneficial to us as humans because it protects our brain and our spinal cord from infection. But when we want to do directed treatments, that is something that hinders us. And then study participants. So a lot of these studies that are in a lot of these clinical trial studies require participants. And the population of Parkinson's disease, even though it's growing, it is difficult to find exactly the patient that fits your study to a T. What I mean by that is, I mentioned that Parkinson's disease has a lot of heterogeneity. So you may have somebody on the spectrum of Alzheimer's and Parkinson's disease, and therefore they are excluded from your study. You may have somebody that appears to present with Parkinson's disease, but is actually not Parkinson's. And so you need to be very careful with who you select for these studies so that you're not getting false negatives, false positives. So I just want to mention the importance of the genetics of Parkinson's disease. And so it's the idea of familial Parkinson's disease, which is the one that can be passed on, versus sporadic. Again, that's that word I use that means it's something that's just developed randomly over time. So the genetic Parkinson's disease, or the familial disease, is actually very interesting and important for researchers to focus in on because this is the one that we can use to track the genetics of it and identify what genes are being turned on or off or being dysregulated that are leading to Parkinson's disease itself. When we talk about the sporadic disease, as it is developed randomly over time, we can't pinpoint why. But when you have familial or genetic Parkinson's disease, that can be used to identify those genes that are being passed down over time and leading to the disorder. And so I've highlighted a few here. You may or may not recognize some of these short forms. The ones in pink are kind of the hot topics right now, and they're autosomal dominant. What that means is if you have a parent that carries a mutation in that gene and a parent who doesn't, about half of the offspring are going to inherit the mutated gene. And even just having a heterozygous copy, which means a copy of the mutant and a copy of the healthier, the non-mutated one, is enough to lead to Parkinson's disease. So an autosomal dominant mutation that leads to a disease is very damaging, as opposed to an autosomal recessive where you actually need both copies of the mutated gene for it to elicit a response. And so the probability of that happening is far less than the autosomal dominant. And that's why something like SNCA, which is the gene for alpha-synuclein, is so interesting. Because only one parent needs to have the mutation, and therefore the odds of actually receiving this is very high. But I should mention, though, that most Parkinson's disease is sporadic. So it's something that happens over time due to aging, which is a completely misunderstood process. Only about 10% of Parkinson's disease cases are familial or genetic. And so the pool of patients that clinicians or researchers can look at and analyze their genetics is very small relative to the overall pool of all patients available. So I'm going to have a few of these images up throughout the slides. Believe me, I don't fully understand them myself, so I'm not going to expect you guys to follow either. But I want to put up, I'm not trying to sugarcoat a lot of things either, because I want to show you that even in these images, which are unbelievably dumbed down, and I don't even understand it, it is still very complex. And so this is the molecular landscape of Parkinson's disease, focusing in on some of those genes that I had listed on the other slide. And I'm just going to highlight the three big ones. So alpha-synuclein, it's very central to Parkinson's disease. This is the one that over time will aggregate and lead to neuronal loss. And then you have this LRRK2, or leucine-rich repeat kinase 2, again, another large enzyme in the body. This one, when it becomes dysregulated, feeds directly into the alpha-synuclein pathology. So a lot of these, if you follow this schematic, you'll see a lot of it feeds into alpha-synuclein. That's just for simplicity. If we were to expand this picture much larger, you would see that in isolation, mutations to any of these genes can lead to Parkinson's disease in alpha-synuclein independent ways as well. And then another one we'll highlight as well is this GCase. And I just want to bring these up because I'm going to mention some of the emerging disease-modifying therapies for Parkinson's disease. So probably one of the more, I would say two of the more popular treatments right now for Parkinson's and other neurological disorders would be one, deep brain stimulation, which is probably better for later stage Parkinson's disease management when levodopa maybe isn't being tolerated as well. And then, like I just mentioned, levodopa is another one. And so this is a dopamine precursor that when your body is no longer able to produce it because you have neuronal loss, you will treat with levodopa. Now that's actually not a disease-modifying treatment, however. That's a disease maintenance. So that is a quality of life treatment. When we talk about therapeutics and disease-modifying therapeutics, we're talking about small molecule compounds, antibodies, whatever these pharma companies or smaller labs can come up with that can actually stop the disease in its tracks. So either stop it before it happens or stop it from progressing. And this is something that does not exist yet for Parkinson's disease or really any other major neurological disorder. And so I'm going to focus on alpha-synuclein right now really quickly. It's a protein that, even though I keep saying it leads to neuronal damage or loss or death, it only does that under adverse conditions. But alpha-synuclein is actually extremely important naturally. It's expressed in almost all of our tissues. It's just expressed to much higher levels in the brain. And what it does naturally is it mediates ultra-fast membrane dynamics. And what that means is, if you think about the role of a dopaminergic neuron, or any neuron, a neurotransmitting cell, is it is constantly shooting out its neurotransmitters. And what's happening at the molecular level is these cells are constantly changing the shape of their structure. So they are bending and breaking their plasma membrane. And to do that in a rapid way, you need something like alpha-synuclein. Now, it's when alpha-synuclein goes wrong does something like Parkinson's disease develop. And so alpha-synuclein in its wild-type form can be shown on the left here to be in kind of two different states. The main state, though, is this, what we call a tetramer. So it binds to itself and it's nice and stable. It's not doing any damage to the cell. Now, when it's mutated, which is one of the more common ways that Parkinson's disease is developed, alpha-synuclein actually forms these fibers, which I've mentioned. And the structure of these fibers can differ, though, depending on the mutation. And so this is important to know because when you're developing therapeutics that can actually target alpha-synuclein, you need to understand its structure. And so the fact that alpha-synuclein, depending on the mutation that some individual may have, can take on a different structure can drastically affect the therapeutic course that you decide to take. And another thing with alpha-synuclein is it's not only intracellular, meaning within the cell and maintaining cell activity, it actually spreads amongst neurons. And that's, again, a normal function. But as it aggregates under a disease state, when it spreads between neurons, you actually get a prion-like effect. And if you know what a prion protein is, it's like an infectious protein that, as it goes amongst cells, it seeds new cells and leads to fibril or aggregation And it does that, and it continues to spread. So even if one cell is the only one that is affected, alpha-synuclein will find its way into neighboring cells and start infecting them. And so these are things we need to consider when we develop compounds that target something like alpha-synuclein. And so I'm going to break down some of the recent advances and some ongoing clinical trials that are looking at alpha-synuclein. So the first one being immunotherapies. So this is developing antibodies that target alpha-synuclein. The idea here is, can we catch alpha-synuclein as it leaves the cell before it enters a new cell and seeds it to begin aggregating and lead to kind of the spread of neuronal loss. And so many of these companies actually have a lot of interesting compounds in various phases of trial. One I want to highlight, though, is this Biogen right here in green on the bottom. And this is really interesting. This is something that was just, I would say a few weeks ago, it was announced. This Alzheimer's treatment for a beta amyloid antibody. It's the first novel therapeutic for Alzheimer's disease since 2003. So we're talking about 20 years. And I know it's for Alzheimer's disease, but what this does is it sets the stage for Parkinson's disease. The fact that it can be done there means it can be done here. And so it's really kind of reinvigorated the community, the research community, in looking at ways to design these antibodies now to target alpha-synuclein, seeing as it seems to be something that can work in Alzheimer's disease patients. Another possible route is aggregation inhibitors. So not only does alpha-synuclein spread, but it aggregates, and it's that aggregation that leads to neuronal loss. So can we design inhibitors that prevent alpha-synuclein from aggregating? We don't want to eliminate alpha-synuclein, but we want to stop it from forming the bad oligomers that we don't, that can lead to neuronal loss. And that's another example of an ongoing clinical trial there. And then cell and tissue therapy. This is an extremely hot field. I'm not very well attuned to this type of biology, but the idea here is when you've developed neuronal loss, it's pretty much already too late, and that's unfortunately the reality of the disease at this stage. But can we develop a tissue therapy that we can introduce to a patient to replace that damaged tissue with new non-damaged tissue? So even if you've developed that damage that is irreversible, you can just replace it with new cells. And so this is an idea that's being looked at in phase 1 right now. Seems promising. To be frank, I wouldn't expect this exact trial to be successful, but the idea here is you have these companies investing in research like this, and it really is just a matter of time for something to stick. And the last one I'll say is this genetic intervention. So there's an idea that you can, okay, alpha-synuclein is bad, at least when it goes bad. So why don't we just introduce some gene therapy that just eliminates the gene entirely? Well, you'll notice that there's no, at least here, reported clinical trial for this type of intervention. And that's because, like I mentioned before, alpha-synuclein is needed by these cells. So even if you want to just obliterate it from tissues, that would actually do more harm than good. And so there's always different types of interventions that, depending on the system you're working in, just don't make any sense. And then I'm just going to quickly go through LRRK2, because I'd imagine some of you in this room have probably heard about this protein as well. It's probably a little bit more hot in the research community right now than alpha-synuclein is for Parkinson's disease. So this is a very large enzyme, and it's important for regulating what's called autophagy. So it's the programmed cell, it's what the cells use to program the recycling of cellular components that have gone bad. So over time, on the millisecond or second or hour scale, within your cells, certain components go bad. Your mitochondria starts to go bad, and they just need to be recycled. It's completely normal. But when autophagy stops working, it's one of the major reasons for Parkinson's disease progression. And so this LRRK2 enzyme is involved in this pathway, and so we wonder if in Parkinson's disease, I should mention that in Parkinson's disease, this protein LRRK2 is heavily mutated, and it's gain-of-function mutations. So what that means is when the protein is mutated, it actually is more active. And so this increase of activity, the cells don't respond well to it. And so what happens is you get something like Parkinson's disease progression. And so there's companies looking at this right now with some clinical phase trials. One's in phase 3. It's actually an extremely late phase trial that seems to be doing pretty well. And the idea here is inhibiting this enzyme when it's mutated so that we can bring its activity back down to basal levels and prevent the progression of Parkinson's disease in that aspect. Now again, this would only be a successful treatment for individuals with Parkinson's disease that have a mutation in LRRK2. There are many, if not more, cases where Parkinson's disease occurs without mutation to this protein. So this is very specific treatment options depending on the individual. And then one of the last targets I'll mention, again, the list of targets is huge. I'm only going to go over some of the hot ones, is this GCase enzyme. This one, again, is involved in kind of that programmed recycling within the cell. This time it's called the lysosome. It's very similar to autophagy that I just mentioned with LRRK2. The idea here, though, is the opposite from what I just mentioned. This enzyme in Parkinson's disease is actually mutated and it leads to a loss of function. So mutation now decreases its activity. And in this case, the decrease of activity leads to Parkinson's disease onset and progression. And so what we want to develop in this case are therapies to increase its activity back up to normal levels. And so it's not, and what I'm getting at here is it's not a guarantee when you have an enzyme that its increase is always bad or its decrease is always bad. It depends on the enzyme, it depends on the context. And any deviation from homeostasis can be bad, whether it's up or down. And so these clinical trials are really interesting. They're gene replacement therapy from Eli Lilly on the left, where it's the affected individual already has a mutated copy of this enzyme, so there's really no changing that. But can we introduce the unmutated or wild type copy to restore the activity? And then also this other company, Bile here, they're developing a small molecule activator. So if we can deliver a small molecule into the tissue that can bind onto this protein, it can bring its activity back up and hopefully ameliorate the disease. So we'll look back at the molecular landscape because we're going to get into a bit of what I've been working on, and I'm going to go through it relatively quickly. But you'll notice here that in this panel, you have mitochondrial dysfunction. And this is, as much as alpha-synuclein is central to Parkinson's disease, mitochondrial dysfunction is also, and I'll mention why, but I also want to bring up, just for context, two well-renowned professors in Canada, both at McGill, who have really set the stage for studying mitochondrial dysfunction and how it relates to Parkinson's disease. And they really put us on the map when it comes to this specific pathway. And it was really, a lot of the work that they did was the foundation for what kind of led into my project as well. And so I'm going to mention first the lab I work in. So I'm local. I'm at the University of Windsor. I was born in Windsor, moved to Amherstburg. Now I'm back in Windsor. What we study in our lab, the Tong lab, is the structural biology, biophysics, and cell biology of proteins. So science is a very broad field. It covers almost anything, you name it. And so I just want to differentiate myself from maybe some others in that we look at proteins in isolation. So to be frank, I'm not a biologist, so we'll see what kind of questions you ask me later. But what we do focus on are any of these interesting enzymes that I've just mentioned, can we isolate them from cells and look at them basically in a test tube? So can we simplify the system down to a couple of enzymes at a time and see how they behave with each other? The specific system, and I'll mention this again, that we're interested in is this human ubiquitin proteasome system. A lot of random words, it doesn't really matter. But what I'm getting at is it takes a lot of different types of techniques to put together a story as to how these biochemical systems work when it comes to the progression of diseases like Parkinson's disease. So somebody like me who's a protein biochemist needs to collaborate with a cell biologist because I don't know what I'm doing when it comes to that kind of stuff, and vice versa. And so something I learned early on, I should say probably a little bit later than I should have, is you can't know everything. So as a younger scientist, you're really interested in trying to know everything, do everything. You want to do it all on your own so that you get your hands on it. And you realize when you try to do something like that, you just get underwater really fast. And so it's important that you kind of find your niche and you stick to it, and you kind of master that craft, and then you collaborate. Because collaboration, without that, there's no way you can succeed. So I'm going to slow down because I know I've been throwing a lot of strange words at you. So I just want to set some foundational principles and mention kind of what we do with biochemistry. So what is biochemistry? It's looking at the nanoscale of a complex environment and simplifying it. So the cell, as depicted here, this kind of crude model of a cell, it's just a really clustered mess of biomolecules, mainly proteins, interacting with each other. What's happening in this complex milieu, we don't really know because there are so many interactions on a microsecond scale, you couldn't possibly keep up with it. So what you want to do instead is look at these things in isolation. And when you look at them in isolation, then you can start to build complex pathways and overall systems like this. And again, you couldn't even ask me what a square inch of this means because there is so much information in this image. But it's the relationship between these biomolecules and how they differ in healthy conditions versus unhealthy or disease states. That is what we're studying. And so I'm going to go over some principles before I get into some of my research in terms of biochemistry. And I'll just read this verbatim. And if you've taken a science course in your time, I'm sure you've heard this before. So maybe I'm just kind of refreshing you on it. But the basics of biochemistry is that DNA holds the information that proteins need, that cells need to produce protein. And proteins are the functional units of life. So proteins do the bulk of the work in the cell. And for all intents and purposes for this talk, we'll just assume they do everything. Proteins are polymers. So when a DNA sequence is read and a protein is produced, the protein itself is made up of many building blocks of what are called amino acids. And so as these amino acids are linked together, you start to build a protein. The order of these amino acids, or these building blocks, is what defines a protein sequence. And so as I mentioned in the slide here, it could be tens of amino acids or thousands. Obviously, the more amino acids you have, the bigger your protein is and the more complex it is. So the sequence of those amino acids, or how those building blocks are arranged, is actually directly related to the structure of the protein. So this figure does a pretty good job at it. It shows the transition from DNA to protein. And as the protein is being synthesized, it's linear by its building blocks. But as these building blocks start to grow, it becomes structured and it takes on a three-dimensional shape. So proteins have these three-dimensional shapes, and these 3D structures are directly related to function. And so the big takeaway here is that structure equals function. So if we know the structure of a protein, we can predict, or based on prior knowledge, know for a fact what its function is. And when you know the structure of a protein, you can do things like make compounds that can target it, make inhibitors, activators, antibodies, stuff like that. And this is really the foundation to any clinical trial. Before anything goes to a clinical trial, these companies are all considering this central principle: is what is the structure of the protein that we're interested in? Without the basic understanding, you can't possibly make it to even phase 1. A lot of these principles too are very much behind the scenes information. What you see in clinical trials is very advanced level stuff, but the years before any clinical trial can take place has to go through some of these fundamentals. And so the interest with the work that we do in our lab at the university is on this ubiquitin proteasome system. And I'll just summarize it as simply as I can. What it is within the cells, proteins are actually modified continuously. So they aren't just existing after they are made from DNA to protein. They don't just exist until they're degraded. They're usually modified in many different ways. And one of those ways is by adding this tag to them. And so this ubiquitin tag is just a kind of a flag saying, hey, degrade this protein, or hey, have this protein move over here and do another function. And so what we're specifically interested in when it comes to this pathway is the reverse of it. So we don't focus so much in our lab on how the proteins are tagged. We focus on how that tag is removed, because the signal from the removal of that tag is actually extremely significant. And there's a bunch of enzymes that actually control that. There's about 100 of them divided into 7 families. So the 7 families of these enzymes shown here. And so one of the enzymes that I'm interested in, and you can't even see it based on the list of all these enzymes, is USP8. And I'm going to talk about what that is. So when we look at mitochondrial dysfunction with Parkinson's disease, something that always comes up in the research is the role that these USP enzymes have. So you'll see USP8, USP30, USP15. So I'll just show you here, USP8, 15, and 30. So these USP enzymes are always popping up, but people, a lot of times, are just kind of disregarding what they do. Maybe I'm not giving justice to all the work that has been done on them. But a lot of times it's just a means to an end. And so our lab is interested in these enzymes and how they can affect something like Parkinson's disease. And you can see in this case that these enzymes are actually either leading to efficient mitophagy, which is the recycling of mitochondria, or they can also lead to the halting of it. And depending on the way you look at it, it can be good or bad for the progression of Parkinson's disease. So USP8 is the one that I focus on. So again, you can't, at least in the years that you do a PhD, you can't possibly look at even all of the proteins in this picture. But if you pick one, you have enough time to hopefully characterize that enzyme and provide the community with some interesting data. So USP8 is the one we're interested in because it's directly involved in this mitophagy pathway, and this mitophagy pathway is directly involved with the progression of Parkinson's disease. So USP8 in mitochondrial health is very complex. The one thing I'll say is it's an enzyme that is very highly expressed in neuronal tissue. So that kind of sets the stage for its importance in Parkinson's disease. The reason I say it's complex, though, is depending on who you ask, they'll either say it is a protective agent against Parkinson's disease, or it's actually something that causes the progression of Parkinson's disease. So there's two sides of the story here, and we're trying to understand kind of where the truth lies. And this is the unfortunate reality of research, is sometimes, depending on the system you're working in, or depending on how you look at a pathway, you can actually see a completely different outcome. And it takes a long time for some of this research to kind of converge and lead to a coherent answer. As it stands now, it's very incoherent. What USP8 is doing is completely unknown. And so we want to step back and ask, okay, how is this enzyme being regulated? So we're not going to focus too much on Parkinson's disease specifically. We just want to focus on how is this enzyme being regulated so that we can hopefully converge some of these ideas and discrepancies. So to do so, what you can do is look at what we know about an enzyme like this in other diseases. So it's really complex in Parkinson's disease, but it's much more well understood in something like Cushing's disease. What we know with this disorder is that USP8, this big protein that's depicted here, kind of in a very simple diagram, is in Cushing's disease, what happens is USP8 is actually mutated in such a way that you get this smaller fragment that is extremely active. So we can measure that activity in the lab, and it's very high. So that catalytic fragment, the activity is about 10 times more than what you see from the non-mutated or wild-type protein. And so that's very interesting because it suggests that the unmutated protein, that entire length of the protein, is regulating itself. And if you cut off the kind of that front, that first half of the protein here, it's unregulated and the activity is extremely high. So that very first half here seems to be doing something. So we're looking into that right now. And one of the things that we can do without even having to step into the lab, is use artificial intelligence to estimate the structure of this enzyme. And if you look at the protein using artificial intelligence and predictive modeling, you get this mess. What we call is spaghettification. It just looks like a bowl of spaghettis. But what we can take away from this is the protein has 3 individual domains. So meaning there are 3 portions of the protein that are well folded. And you can see the shape of those domains shown there. How those domains interact with each other in the context of the entire protein, though, is completely unknown. And so we want to characterize that, and that will be the key to how this enzyme is regulated. So there's a whole bunch of ways that you can do this. All of these fancy acronyms are shown. And these are just different ways to look at a protein at different scales. So you look at the atomic scale, so where are those atoms, the carbons, hydrogen, nitrogen, oxygen. Or you look at it more at a shape scale, which is what we ended up doing. So is the protein compact, like this picture indicates, or is it maybe elongated, and is the artificial intelligence completely wrong? So we use this technique called SACS. I'm not going to get too much into this. It's very much a biophysical technique. What I'll say about it is it's a shape technique. So it's a method of, I guess I'll mention in the next slide, but it's a method of understanding the overall shape of your protein. So it's what we call low resolution, but you can learn a lot about the shape. To do an experiment like this, you actually have to travel. Luckily, because I'm in Windsor, not very far. So we just go about 3 hours to Illinois. And what we're doing is we're going to a synchrotron. So this is a particle accelerator. This large ring, it's about 1 kilometer in circumference. It's accelerating electrons at relativistic speeds, so meaning near the speed of light. And when that happens is, what happens when you do that with electrons is they actually release X-ray radiation. And X-ray radiation is the perfect wavelength for looking at protein or molecular structures. So at this beam line, as we call it, or at the synchrotron, we can bring our sample, and this is just a video of the automated sample handling at an institute like this. What this robot is doing is it's putting the sample in the front of the X-ray radiation beam. And when the protein is exposed to X-ray radiation, you get something that looks like this. Honestly, kind of boring. It doesn't really mean much to me either. It's a scattering profile, though, and there's a ton of information in here that the human eye can't see. But when you break it down using mathematics and physics, you can actually understand the shape of your protein at a kind of a global scale. And what we found is that USP8 is elongated, and it's kind of like this long tube, this kind of sausage shape, whereas the artificial intelligence was suggesting that it's actually more compact. So it turned out that the AI estimates were way off. The reason this is significant is it suggests to us that the shape of this protein, namely these distant domains, are likely not folding over. So these distant domains here, they're likely not folding over and interacting with the other distant domain. It's got to be some short range interaction. And so we wanted to characterize how this enzyme is regulated, and we realized that there's a really small domain that people haven't realized before that is folded, and this is acting as the inhibitory unit. And so when that domain is present in a unmutated USP8, the enzyme activity is relatively low. But when you take that domain away, so that delta WW there, the activity increases significantly. And so it shows us that that little region of the protein is what is regulating the enzyme. And so we actually figured out what the structure of that little tiny domain is. I'm not going to talk much about the method here. And we were able to characterize this domain using various techniques to show that it is indeed what we thought it was. And if you do just a single mutation of that domain, so instead of taking away the entire domain but do a single mutation, what you're left with is, again, an enzyme that is far more active than the unmutated protein. So again, it's more information suggesting that we have indeed a regulatory unit. And so we did a little bit more in terms of the biochemistry, looking at how this is regulating the enzyme. And what we found is that this WW, this small regulatory domain, is interacting with the catalytic domain simply because it is tethered to the protein in such a way that it's flexible enough to kind of move around back and forth. And really quickly, what I'll mention is we don't quite know how this is happening. It's something that we're currently working on to determine exactly what is going on at the atomic level. And then I probably won't get much into this just because of time, but we did characterize a bit more on what this domain is doing in terms of regulating the protein at more of a physiological level. It turns out that it's actually modulating the enzyme's activity using binding partners. And so this is where these pathways start to grow as you add more and more proteins to your system. So I don't really have much time to go over this, and I think we're all happy I don't either because of how complex it's getting. What I'll say is it's a bit more biochemistry, kind of feeding into that understanding of how the enzyme's regulated. And again, a little bit more of this as well. And the take-home message here, again, is kind of this image at the bottom, is that this enzyme is being regulated by this small domain purely based on the protein's dynamics. So we understand now the protein structure, and we know its function. So that was kind of the idea here. So the conclusions are that USP8 is very extended, and that the regulation of this enzyme that is likely linked to Parkinson's disease is being mediated by this really small domain. The future directions for this project, now that we understand how the enzyme is being regulated, is to look at it in the context of Parkinson's disease. So how does this enzyme now regulate something like alpha-synuclein? How does it regulate mitophagy? Because these are both pathways that it's directly involved in, and these pathways are both directly involved in the progression of Parkinson's disease itself. So I'll just quickly say some acknowledgements about the lab. There's Dr. Tong there, he's the supervisor. Some of the lab members, graduate students at the top. We have some undergrads at the bottom. All the collaborators, a huge amount of effort with some of these experiments. And of course, I want to thank the funding institutes, especially the Parkinson's Society, for kind of getting us onto this project. It's been a year-long project of understanding how this enzyme works, but it was definitely well worth it, we find. Thank you.